Good morning. Is that working? Yes. So, good morning for this first plenary lecture. Welcome to Glasgow. I'm Danny Hoyer from Melbourne, and you should all have something like this caller to go to Melbourne in 3 years from now. Right, so I will talk closer to the microphone. It's my great pleasure to introduce you to Professor Ed Bullmore for the first lecture. So this is Inflammation and Depression, just in case you wonder where you are. So Ed was trained in medicine at the University of Oxford and also in London. Then he did psychiatry at the Maudsley. Moved to Cambridge as a Professor of Psychiatry in 1999, head of the department from 2014 and 2021. He's currently director of the Wolfson Brain Imaging Centre and deputy head of the School of Clinical Medicine. He's also honorary consultant psychiatrist and research director at Cambridgeshire and Peterborough Foundation of the NHS Trust. From 2005 to 2019, he worked half-time in GlaxoSmithKline. I have to say I worked with Novartis for 31 years, so welcome to the club. As Vice President of Experimental Medicine, and then it focused on immuno-psychiatry as described in his best-selling book, The Inflamed Mind, which was published in 2018. So Ed has published at least 500 papers, and we stopped counting. His age index is 175, and he's been elected to a number of learned societies and boards. He's a fellow of the Royal College of Physicians, Royal College of Psychiatrists, Academy of Medical Sciences, and actually was also the treasurer since 2018. And to cut all things short, Ed, welcome. The stage is yours. This is still working. You need some help with the sound. You're doing it, Danny. Okay. Just bear with me. Be a little bit patient. This is technology, and clearly it has not been tested before. Interestingly, all these work.
You can hear me now? Ah, yes, that's much better. Yeah. Okay, well, look, thank you very much for that introduction, and thank you all for turning up at 8:30 on a Monday morning to hear me talk about depression. That's commendable commitment to the cause. So I'm going to talk about inflammation and depression, some questions of causality. I was thinking about this as a pharmacological conference and thinking I'm not going to say very much about drugs. What I am going to talk a lot about is targets. What do we need to know about a potentially new class of targets, in this case, immune mechanisms for treatment of psychiatric disorders, and I'm going to focus, as I've said, on inflammation and depression. So you've heard a bit about my bio. This is just so that you can see for yourself my employment and sources of research support. And the outline of the talk is really in three parts. I'm going to start by talking about inflammation and depression. I'll introduce it as a kind of forbidden association that nevertheless raises the prospect of a new class of psychotherapeutic drugs. I'm then going to go into the questions of causality. What do we really mean by causality, and how can we begin to verify causality in this context? And in the final part, I'll take a bit of a knight's move out and talk about brain development and what we know about brain development, the factors that impact on brain development, and you will see that it all comes to a hypothetically integrative conclusion at the end. So, inflammation and depression are associated with each other. I would dare to say that that really is beyond reasonable doubt. It's not, or at least it wasn't written in the textbooks when I was reading medical textbooks, but I think anybody that has practiced as a physician or psychiatrist or general practitioner will almost certainly have met somebody like this woman. She's an illustrative example of a patient with a medical inflammatory disorder. She has rheumatoid arthritis. You can see her joints are a little bit deformed. And I put her up there because she reminds me of a patient that I saw when I was training as a physician in a rheumatology clinic who had joint pain, nodules, various other physical signs and symptoms of rheumatoid arthritis, and she looked unhappy. And I talked to her, and she was clearly depressed. I went to the consultant in charge of her case and said, you know, this patient is not just suffering from rheumatoid arthritis, she also has depression. He looked up briefly and said to me, well, you would be depressed, wouldn't you? And that was the end of the story. She received no treatment for her mental health symptoms from us in that rheumatology clinic because the assumption at the time was psychological symptoms like depression, fatigue, anxiety might co-occur with rheumatoid arthritis, but they're not really part of the physical disease mechanism. I'll come back to that in a minute. Now, this kind of blind sight that we have, we just don't see, or medically we don't recognize the importance of psychological symptoms associated with physical inflammatory disease, is not because these are rare patients. They're actually pretty common. The chart on the right here is showing you some data from randomized clinical controlled trials that were run by GSK or Janssen. So these are industry standard trials, carefully screening patients with a number of different inflammatory diseases, ankylosing spondylitis, lupus, rheumatoid, etc. And in each trial, an instrument called the SF-36 was used as a general measure of well-being, and that instrument includes two questions, one about low mood and one about capacity for pleasure or loss of pleasure, also known as anhedonia. And what you can see here is that about 20% of those trial patients scored highly on those two symptoms of depression. So even in a trial population, this is quite common. And if you look at the scatter plot on the right, you can see each point here representing a different disorder. The percentage of patients in that disorder group with high depressive symptoms scales positively with mean baseline concentration of CRP or C-reactive protein, an acute phase protein that you'll know is commonly measured in the blood as a marker of innate immune activation. So this is just an example of some overflowing evidence to suggest that inflammation and depression are associated with each other. But I called it a forbidden association because it isn't very well recognized within medicine that psychological and physical symptoms should be caused by the same mechanism. And that fault line, if you will, between mind and body goes back a very long way. I would say it goes back at least 400 years to the time of Descartes and his famous exposition of a dualist philosophy. He defined each of us as humans being essentially made up of two completely dissimilar components. There was a physical machine, the body, which was mathematically analyzable, experimentally tractable, obeyed the laws of physics. And then there was the mind, or the soul, as he called it, which was immaterial, unquantifiable, experimentally intractable. In other words, as different as it possibly could be from the body. Now, we might look at this and laugh at the way that the very beautiful, rigorous analysis of light beams and muscle action represented in this engraving converges unrealistically on the pineal gland as the seat of consciousness shown here. We know the pineal gland is not nearly as important as Descartes thought it was, but his basic idea that there's a fundamental split between mind and body is still very entrenched in Western medicine, and it makes it rather difficult for us to think about things like immune mechanisms for mental health disorders. Now, you might think that this is all just a bit academic. I'm sort of going back to the 17th century and giving you a bit of old philosophy. What's it really got to do with real life as we know it? And I would contend that this dualist idea is actually very much still entrenched in how we think about mental and physical health disorders and how we practice as physicians or psychiatrists. And this is an example. These are the diagnostic criteria for major depressive disorder as set out by the American Psychiatric Association in the DSM-5, the Bible of Psychiatric Diagnoses. And you may or may not be familiar with these criteria, but basically to have a diagnosis of major depressive disorder, or MDD, you need to have at least one of these two core symptoms of depression, depressed mood or loss of pleasure, and some of these other symptoms. And you can just see by looking at the way these other symptoms are defined that a group of patients with MDD is going to be quite heterogeneous. Your sleep could be excessive or insufficient. You could be eating too much or too little. You could be gaining weight, losing weight. You could be physically overactive or retarded. All of those are symptoms of depression. It's extremely inclusive syndrome. However, there is one important exclusion criterion, and that is symptoms must not be attributable to a substance or medical condition. So the patient that I saw in that rheumatology clinic all those years ago ticked all these boxes. She had all the symptoms, but formally she could not be diagnosed as having a major depressive disorder because of the dualist fault line that runs through the middle of psychiatric diagnostic systems. Now, what if we challenge that fault line and find patients with major depressive disorder, take blood from them, and analyze the markers of the immune system that we find there? A lot of people have done this over the last 20, 30 years, and this is a meta-analysis of 4 independent primary studies looking at whole genome expression in whole blood samples from about 1,500 patients with depression and about 1,000 healthy controls. And this plot is just showing you that there are quite a lot of genes that were significantly over or underexpressed in depressed cases compared to healthy controls. You might look at that list and perhaps you can decode some of the acronyms, but it's easier, I think, to use bioinformatic tools like this, which map the gene list of overexpressed genes in depression onto underlying biochemical and signaling pathways. And you can see that a lot of these genes that are overexpressed in depression are functionally specialized for functions such as neutrophil degranulation and other aspects of innate immunity. So when we look at the genes, we see evidence for activation of the immune system on average in depression. When we look at the blood cells, the immune cells circulating in the blood, we see a similar kind of story. These are data from about 200 patients with depression. And we've measured 14 different cell types, neutrophils, eosinophils, basophils, etc., and then used the technique of principal component analysis to summarize shared patterns of variation across all those different cells. And you can see that the patients with depression have increased immune cell counts and actually also increased red cell counts and platelets compared to healthy controls. But perhaps more interestingly, when you just look within the group of depressed cases, you can see that there's some evidence for subgroups, 2 subgroups in this analysis. One is depressed, but with normal immune cell counts. The other, shown in red, is depressed and has increased cell counts, increased CRP, increased IL-6. I'm not showing the protein results on this slide. And also, as shown in the box plots on the right, you can see that inflamed subgroup of patients with depression has higher scores on the HAM-D, the Hamilton Rating Scale for Depression, a physician-rated questionnaire, and the Beck Depression Inventory, which is a self-rated questionnaire. So these are examples of a literature that has grown very rapidly, demonstrating that we can measure proteins, we can measure genes, we can measure cells in the peripheral blood, all of which indicate moderate or low-grade inflammation peripherally in people with depression and suggest that there may be a subgroup of more inflamed cases. How does this work, though? How do we get from protein, cells, and genes in the blood to a state of mind like depression? Well, clearly the brain must be involved, and we and others have begun to integrate neuroimaging into these studies. So this, for example, is a study where we've used a magnetic resonance imaging technique called proton density. It's a structural MRI metric, but unlike the sort of conventional structural MRI metrics in terms of sort of brain volume or cortical thickness, which are sort of macroscopic measures of brain anatomy, proton density is a so-called microstructural MRI marker, which tells us something about the tissue composition within each voxel. In particular, it tells us something about the water concentration within each voxel. And this map is showing that in this part of the brain, which is the posterior cingulate and precuneus, in the back of the brain on the medial surface of the hemisphere, there is increased proton density correlated with increased CRP in these cases and controls. So more inflammation, more water in these areas of the brain measured by proton density. When you think about it, inflammation everywhere else in the body is often associated with extravasation of water, edema. Maybe it's not that surprising that we see a similar signal in the brain associated with peripheral inflammation. If you look in the neurological literature, proton density is quite widely used as a marker of brain edema associated with hepatic encephalopathy, multiple sclerosis, and other such disorders. But it was a new finding in relation to depression. We thought it was a plausible finding, not least because this locus of increased proton density, increased brain water in the posterior cingulate coincided quite neatly with part of the circuitry, the so-called default mode network shown here in blue, that we know is functionally disconnected in patients with depression. So we're beginning to pull together structural and functional MRI data in relation to depression. This is another study in the same sample, focusing in a bit more detail on how inflammation might be related to the functional organization of the brain. Here we're using network analysis to demonstrate a group of cortical areas, shown here as colored nodes, where the connectivity between those areas is affected, attenuated in association with CRP. And you can see that perhaps more clearly in the scatter plot as the concentration of CRP increases, so the functional connectivity in this network, which is specialized for interoceptive function. So this is the part of the brain that basically reads the body, stays tuned to the body, tells the brain how the body is doing. This interoceptive network has reduced connectivity in association with increased levels of blood inflammation. And these plots show you essentially the same data, but through a different prism, as it were. Here we are representing each cortical area as a colored segment around the perimeter of this circle, and the lines drawn between cortical areas on the rim of the circle show the change in connectivity between the group of patients with low immune cell counts that I showed you in a previous slide compared to the group of patients with high immune cell counts. So the more inflamed patients are showing thicker, bluer lines between regions of the default mode network and the ventral attention network, which is compatible with the idea that you've got reduced connectivity in this circuitry. So if you are acquainted with MRI, you will know that in many ways it's a wonderful and very versatile technology, but it is not very molecularly specific. So we can see things like water change, we can see these functional connectivity changes, but really what we want is better measures of brain inflammation that are more precisely specific at a molecular or cellular level. And we have very few such markers available to us. This is one. It's positron emission tomography, PET, using a tracer called PK11195, which binds to the translocator protein on mitochondrial membranes and is often regarded as a marker of microglial activation. I actually think that's quite an optimistically specific interpretation of PK11195 binding, but that's the way it is often construed. And here you see some data from a study that we did showing in patients with depression there is increased binding of this radio tracer, specifically in the anterior cingulate cortex. It's not a very big signal, to be perfectly honest, but as shown in these forest plots, is compatible with the kind of effect sizes that other groups have reported using similar technology. And overall studies, there is a grand mean effect of significantly increased TSPO ligand binding in various brain regions, compatible with the idea that there is inflammation associated with depression, not just in the blood, but in the brain. Now, if you are optimistic like me, you would look at these data and think the evidence for association is so compelling, it must be the case that anti-inflammatory drugs would have antidepressant effects. And actually, there is quite a lot of evidence for that, but it's not entirely straightforward to interpret. And the reason for that is that the evidence we have, the best evidence we have, is derived from studies that were designed for a different purpose. So these are the results of the randomized control trials that I introduced earlier from Janssen GSK, looking at patients with lupus, ankylosing spondylitis, rheumatoid arthritis, etc., medical inflammatory disorders, treated with a range of different anti-inflammatory drugs, many of them monoclonal antibodies. And what we've done here is analyze these trial data as if the SF-36 instrument that I mentioned, this secondary endpoint as it was in the original protocol used to measure patient well-being, we've analyzed those data as if they were the primary endpoint. And what you can see is there's variability in terms of a treatment effect, but again, the grand mean effect overall studies shows that anti-inflammatory treatment compared to placebo has a significantly non-zero effect in the order of about 0.3, which might not sound like a lot, but it is actually quite close to the grand mean effect size of conventional antidepressants like SSRIs. Now, you show these data to, let's say, a more skeptical audience, and they will be quick to say, well, this doesn't tell you that anti-inflammatory drugs have antidepressant effects. It could all just be a secondary reaction. Maybe the anti-inflammatory drugs are improving physical health and mood is improving as a consequence of improved physical health. So not a direct mechanistic effect of the drug, but a psychological reaction to the benefits of drug treatment in the physical domain. A classic dualist interpretation. It's difficult completely to discount that interpretation because the trials, as I've said, were not designed to test these drugs as antidepressants. One secondary analysis that you can do, that we did, that I found was quite persuasive, was to restrict the analysis only to the subgroup of patients in each trial who did not respond physically. So they had arthritis, let's say, but their joint pain did not improve. If we just look at those patients who did not get better physically and ask the question, was there a drug treatment effect on their mental health outcome? The answer, again, is yes. In fact, the ground mean effect size for the patients without a physical response is actually a little bit bigger at 0.38 than for the full data set. So you can believe that anti-inflammatory drugs or immune modulating drugs might have benefits for psychiatric treatment. And a lot of people are thinking about this quite hard, and this is a review that we published early last year, which is interesting perhaps if you want to follow up on the detail. I put it up here not to go into each and every box on these complicated diagrams, but just to emphasize that there are a lot of potentially druggable targets for which there is some evidence that they might be implicated in depression or other mental health disorders. This one, for example, the P2X7 receptor, sits on the surface of microglia and neurons. It's a purinergic receptor under conditions of biological or social stress. High levels of ATP open this channel. Calcium influx activates the inflammasome, and the cells produce pro-inflammatory cytokines like IL-1. So this is a particularly interesting target because it seems to sit on a mechanistic pathway between stress and brain inflammation that obviously might be important for psychological disorders. However, nothing I've said so far really demonstrates causality. And as you'll know, if you've ever done any drug development work, you do not want to be running a drug development project against a target which is a bystander, which is not actually causally implicated in the disorder you want to treat. So how can we begin to move beyond the association that I've introduced between inflammation, broadly speaking, and depression, broadly speaking, and get to something a little bit more causally specific? Well, first, I think we have to clarify that by inflammation, I'm essentially talking about activation of the innate immune system. And when we think about depression, maybe we should think more specifically about particular depressive symptoms: anhedonia, fatigue, so-called atypical depression associated with increased weight, increased eating, increased sleep, brain fog or mild cognitive impairment, sickness behavior, and transdiagnostic depression or depressive symptoms that cut across the conventional psychiatric disorders. All of these have been advocated in the literature as the psychological consequences of inflammation. I've mentioned that we need to understand the role of the brain in mediating these connections, but we might also ask ourselves, if we really want to pursue causality, okay, so innate immunity might have something to do with it, but where does that innate immune activation come from? And here it's interesting to note that many of the obvious causes of chronic low-grade inflammation, comorbid infectious or inflammatory disease, the sequelae of COVID or other infectious diseases, obesity, all of which are correlated with depression, increase the risk of depression, and of course increase the risk of inflammation. I've been particularly interested by the literature indicating that not just biological stresses like inflammation or autoimmune disease, but also social stresses, in particular early life adversity, can cause an immune response, and quite a sustained immune response. And when you think about how early life adversity might lead ultimately through an immune pathway to depressive symptoms, I think you have to invoke the adaptive immune system, the memory cells, the T cells, the B cells of the immune system, as well as the sort of short memory, quick-fire innate immune cells. Why do I think the story about early life adversity is so interesting? Well, partly because if we know anything in psychiatry, we know that the conditions of early life are absolutely crucial for future mental health. And that goes back at least to Freud, probably further. And this is some of the data to suggest that that long memory of early life adversity could have something to do with the immune system. This is a longitudinal study from New Zealand, the Dunedin cohort. They recruited a cohort of kids in the first decade of life, very carefully analyzed their social environment, so they contemporaneously assessed exposure to adverse experiences and then followed them up for many decades. And what you can see is that children who had had more than two 2 or more adverse experiences, were significantly more likely as adults to have depression, to have metabolic risk markers elevated, to have physical disease, and to have increased levels of inflammation indexed by CRP. So these are measurements being made decades after the exposure, and yet they suggest an enduring effect on the immune system. How can we understand that? How can we understand such a long memory? Well, one mechanistic idea that has been around in the animal literature in psychiatry for quite a while is that social stress, particularly early life stress, can become biologically embedded by epigenetic modification of cell genomic transcription. There are multiple different epigenetic mechanisms that have been implicated in this way. I think for many of us, for many years, this seemed like interesting animal lab stuff that was never really going to translate to the clinic because we can't read epigenetic modifications in neuronal cells. But as this slide shows, people are increasingly curious about whether it's useful to measure epigenetic status of peripherally circulating immune cells. Could that give us some insight into how stress might be biologically embedded? And recently, together with my colleague Mary Ellen Linnell in Cambridge, we did an analysis that I think begins to support that idea. And what she did was really conceptually quite simple. She accessed open data on about 80 or 90 different tissue types. And in each case, she looked at the profile across the genome of methylation, and then asked, do the peaks of methylation indicating epigenetic modification, do these peaks coincide with the loci of risk genes for psychiatric disorders, broadly speaking, so-called transdiagnostic GWAS variants, in any tissue type in particular? And what she found, to begin with, quite encouragingly, was that neuronal cells, that was certainly true, particularly fetal neuronal cells. Epigenetic activation coincided very significantly with GWAS loci. That was not true for any other organ or tissue type except in the blood, the T cells, particularly the adaptive immune cells in circulation. They also showed this epigenetic enrichment for GWAS risk variants. It's kind of interesting. She pursued it by finding another independent epigenetic resource, so-called blueprint. Looked again, similar story, measuring the epigenetic profiles, this time using histone acetylation across a range of different cell types, and replicated what she'd previously found. In other words, in T cells specifically, there was significant enrichment of histone acetylation at GWAS loci implicated in transdiagnostic risk in schizophrenia and in BMI, as well as depression. So these and other data begin to point us in this kind of direction. Now, this is obviously a speculative model, but I am quite intrigued by it. And the idea is that, let's say in the periphery, you are exposed to infection, or it could be a social stress. That leads to epigenetic modification of genes. And if you've inherited at those genetic loci, if you've inherited risk variants for psychiatric disorder, then the stress exposure will activate those genes and modify the phenotype of T cells, which may in turn allow low-grade inflammation through myeloid cells, generating the signals that we've been measuring in the blood, and that I showed you earlier, the increased levels of inflammatory proteins in cells. So But these T cells also exist in the brain or can get into the brain across the blood-brain barrier and could then modify neuronal function directly. It's a speculative theory, but it's a very interesting theory from a psychiatric point of view because it draws together several different threads. Not least, it is relevant to the growing evidence that the immune system is crucially important for brain development. Synaptic pruning, for example, this loss of synapses that is part of normal maturational change in the brain, we now understand may actually be directly done by microglia and other phagocytic cells in the brain. They may simply eat synapses during the course of development. So you have an immune cell very much implicated in brain development, and then you've got this evidence emerging that stress exposure could, through epigenetic mechanisms, modify the phenotype of immune cells, potentially having an effect on brain network development. Which brings me to the third and final chunk of data that I want to just go through briefly with you today, and that is more about brain development. And what do we know about that, and how might it all tie up with what I've told you so far? So the first thing to say about brain development is that we know a lot more now than we, I think, have ever done because of open science. Open science has been a huge boon in this area, and this study that we published last year was a beneficiary of that trend. So we found accessible data on about 120,000 brain scans from individuals right across the life cycle, from before birth, so fetal MRI scans, all the way through to the late 90s. You'll notice the age range here, by the way, is on a log scale. So what we're seeing is a lot of activation, a lot of activity, increasing grey matter volume, increasing white matter volume, particularly in pregnancy and in the first few years of postnatal life. This is the crucial period of brain development. And the brain remains plastic throughout adolescence and into early adulthood. All of this coincides, of course, with the period of peak risk for incidence of mental health disorders. So the idea that mental health disorders have something to do with brain development is a very natural one. But what we mustn't do is start thinking or talking about the brain as if there's one canonical central nervous system, when in fact, as we get more data and as we think about it in a more, let's say, epidemiological way, it's clear that there are a lot of important individual differences. So this is a rather busy slide which looked at functional connectivity changes during adolescence and focused specifically on whether there were sex differences in how the brain changes in terms of its functional connectivity during adolescence. And we found that there were indeed a set of regions, cortical and subcortical nuclei, where functional connectivity in women specifically developed quite strongly during adolescence. We call that the female more disruptive system, shown here. And this system turns out to involve brain regions that are important for reward, motivation, other psychological functions that you can imagine might be relevant to depression. It was spatially co-located with the pattern of disconnectivity, functional disconnectivity in the adult brain of patients with depression that I showed earlier. So these adolescent girls are showing an accentuated developmental change in functional connectivity of a circuit that we know is abnormal in adult depression. And we can link the imaging data to transcriptional data, gene expression data in the Allen Human Brain Atlas, and find the genes that have an expression pattern co-located with the imaging phenotype of disrupted or disruptive functional connectivity development. We can find the genes that most look like that MRI phenotype in terms of their spatial expression pattern, and we can demonstrate that those genes are enriched for depression, suggesting that this sex difference in development of the brain could be relevant to the sex difference in incidence of depression, much commoner in young women, as you know. So sex is an important individual difference. The genetics of psychiatry is a very exciting story. You know, for years and years, we knew that mental health disorders were heritable, but we didn't really know which genes were important. Now we have GWAS studies that are of sufficient scale to identify specific genes. We can calculate polygenic risk scores, and we can relate polygenic risk scores to brain structure and brain function. And here is an example of that approach, demonstrating that these areas of the brain shown in the bottom right-hand panel are correlated with polygenic risk for schizophrenia. If you've got a high genetic risk for schizophrenia, you will have less of a microstructural marker called neurite density index, a measure of connectivity at a microscopic scale in these brain areas. So sex is important, genes are important, and environment's important. These are data, again, looking at adolescent development. These are the areas of the brain that are strongly myelinated in red at the age of 14, the early myelinating areas of primary motor and sensory cortex, and then here you see in yellow the later maturing, later myelinating areas of association cortex. And what we found in this study was that kids who were in the lower third of the distribution for socioeconomic status compared to kids that were more affluent and been brought up in families in the top third of the socioeconomic distribution had different rates of maturational change, particularly myelination was different or occurred at a different rate in this frontostriatal circuitry. So brain development, it's all happening in the first, really a lot of it's happening in pregnancy and in the first few years of life, but it goes on happening throughout adolescence, and it's subject to genetic, environmental, and sex differences. How can this all be relevant to what I told you to begin with about immune mechanisms? Well, this is, I think, a very interesting integrative theory. You can see from the diagram, it's quite broad brush. It's a bit of an artist's impression, this diagram. It's not my theory, but I think it's very interesting. It's called the neuroimmune network theory, and the idea is that when we're exposed to early life stress, or actually adult stress, but let's particularly focus on the early life story, there is a whole body reaction to that event. It involves the sympathetic nervous system, the HPA axis, the immune system. And the evidence from epigenetics implicating adaptive immune cells in the memory of that event is quite compatible with this theoretical idea that you have then for long-lasting, low-grade peripheral inflammation, which generates some of the signals that we read off in blood samples, increased CRP, increased IL-6, but may also modify brain development to the point that the adolescent or adult ends up with a slightly atypical brain organization that confers increased risk of behaviors and cognitions that ultimately we recognize clinically as the symptoms of mental health disorder. It's the neuroimmune network theory. I think it's a very interesting integrative idea. So I'll finish with a few take-homes. I've tried to persuade you that the association between depression and inflammation is beyond reasonable doubt, but there are important questions still remaining about causality and specificity of that relationship. Epigenetic activation is theoretically interesting, and we now see a practically accessible mechanism for a long memory of early life adversity that's going to be an important story, I think, in the pathogenesis, not just of depression, but pretty much all psychiatric and neurodevelopmental disorders. We also are increasingly aware that during the time of greatest risk for mental health disorders, the brain is in a very active state of development. And then we've got these neuroimmune integrative ideas. And I've put my version of the neuroimmune network theory in the bottom there. Early life adversity is biologically embedded by epigenetic activation of risk genes in immune cells, as well as nerve cells, leading to atypical development of neurocognitive networks and long-term negative health outcomes. So there's plenty of work still to do to explore those ideas in more detail. I want to close by thanking the many people who've done the work I've briefly presented, to shamelessly promote my book, The Inflamed Mind, which is a general guide to some of the ideas I've mentioned, and also the textbook of immuno-psychiatry, which provides a bit more detail for those that are interested. And with that, I'll finish, and thank you again for your attention.
It was a fantastic talk, Ed. You convinced me that DSM-5 needs to be updated, at least.
Yeah.
Now, we have time for questions, and of course I take the privilege to ask the first questions anyway. So is long COVID the perfect illustration for what you're trying to tell us?
Yeah, in some ways, yes. I mean, you know, there is a sort of, I don't particularly like this phrase, but you can think of COVID as a natural experiment. And, you know, a huge chunk of the population was more or less simultaneously exposed to this pathogen. We know there were a lot of brain consequences of COVID, particularly in the first waves. And then we've seen this long COVID phenomenon. Now, long COVID is protean, you know, so in other words, different people will mean different things by long COVID. And it doesn't necessarily align perfectly with DSM categories. But we and others have shown, or are in the process of generating data, to suggest that a lot of the neurological and neuropsychiatric complications of COVID are to do with the immune reaction to the virus. You may remember when COVID first emerged, there was some anxiety. It is a neurotropic virus. There was some anxiety that would actually get into nerve cells, perhaps in the nose. A lot of people had anosmia, track back through the nose and get into the brain and directly infect nerve cells in that way. It seems it's unlikely that that's what it does in humans, at least genetically intact humans. But what we do see much more clearly, both in animal models and from clinical studies, is that the immune response to the infection, both in the brain and peripherally, is very strongly coupled to fatigue, depression, anxiety, cognitive impairment, a lot of these psychological symptoms that form part of the long COVID syndrome.
On another note, we've been working with the ligand, the PK111, I mean the translocator protein. Yeah. I wonder, once you have patients that have been imaged before treatment and after treatment, do you see changes in the signal?
Well, we haven't done that experiment. And I'm racking my brain to think if anybody has done that experiment, and I don't think they have. I mean, I tell you what, this is a little bit left field, okay, but, you know, one of the things that intrigues me, or has intrigued me most recently about the PET literature, is people say, we haven't got very good markers for brain inflammation. Do we need a different kind of ligand? Is there something that's more specific to the microglia? And that's a perfectly legitimate sort of line of inquiry. The other angle that has cropped up recently is the idea that actually we should perhaps be looking at immune compartments in the head that are not necessarily in the brain, that might give us a stronger signal and might be relevant to neuro and psychological complications of inflammation. So measurements of the bone marrow in the skull, or the immune compartments in the meninges, or the sinuses. You know, there's a lot of tissue in the head which is actually home to the largest concentrations of immune cells, not so much in the brain. And I think it's very interesting to think, you know, could we be using PET, maybe even existing PET data, to look at those intracranial but extracerebral immune compartments, and could those be relevant to neurological disorders? Maybe we don't need a new ligand, maybe we just need a different way of looking at the data we've already got.
Well, we certainly can do a network analysis if you've done, and you can see that the default mode network, I mean, all these communications were completely cleaned up. Are they, oh yes.
Phones aren't working.
It works now, Mitra.
It is now.
Yes.
Hi, I'm Rita Alawali from Queen Mary, University of London. I very much enjoyed your lecture. You spoke a lot about activation of immune pathways, and they're pro-inflammatory in nature. But in one of your slides, you showed minocycline and the possible pathways that might underlie some of the benefits.
Yeah.
And one of those was anti-inflammatory. So in other fields, such as in coronary artery disease, there's this growing belief that the inflammation, which is critical to disease pathology and development, that it's actually the resolution pathways that are impaired.
Yeah.
It's the turning off that is an issue. And I was looking in your genes and also all of your large-scale analyses, and I didn't really see too many pathways that might say, rather than activating inflammation, it's actually a loss of the turn-off of inflammation. Have you looked at that?
No, we haven't actually specifically looked at that. And you're right, the field is very focused on the kind of pro-inflammatory drivers rather than the anti-inflammatory modulators or controllers of inflammation. And this idea that the better target might be the one that helps inflammation switch off rather than preventing inflammation going in the first place. I mean, that's certainly a very interesting idea. I think there are people thinking about Tregs, for example, which I guess gets you into that space. So there are colleagues at Manchester who are quite fascinated by the idea that Tregs, which you could think of as perhaps adaptive immune cells that might turn off some aspects of innate inflammation, are implicated in schizophrenia. That's their hypothesis. And they are exploring the possibility of doing a repurposing study of methotrexate, which apparently has some effects on Treg cells that might be compatible with the line of questioning that you've articulated. But I think what I would say is what I would say in general is that we need more immunology expertise in this space. You know, the immune system is as complex as the nervous system. And when you start talking about immune mechanisms of mental health disorders, you're really talking about a lot of intersecting domains of complexity. And we do need to make sure that the field isn't dominated by, psychiatrists and neuroscientists who are a little bit naive or underinformed on the immune side. If there are people in the audience or in your networks with immunological expertise who want a next generational challenge, then please think about mental health. Please think about neurological disorders as well, because there's a whole big story about neuroimmune mechanisms for dementia and various other neurological disorders. And in all these areas, we need strong immunological expertise coming in to make it really work as an interdisciplinary science.
And I would suggest to you that it's probably really important, because this is I suppose you'd call it chronic disease, and long-term suppression of the immune response, so suppressing our ability to fight when it's needed, is probably not a good idea in the long term. And in fact, in cardiovascular disease, the idea has been tested, and whilst it seemed to have benefit, the risks outweighed the potential benefit.
Sure. Yeah. That point is well taken.
Good morning. Good morning.
Good morning.
Doctor, thank you for that wonderful presentation. In the light of what you have presented today, do you think that there's a need to re-evaluate how depression and further mental disorders are diagnosed or evaluated? Should we be rethinking the DSM categorization? Thank you.
Yeah. Well, I think we should, and a lot of academic psychiatrists like me have thought we should be rethinking DSM for a long time now. Tom Insel, who was the director of the National Institute of Mental Health for quite a long time, produced something called the Research Domain Criteria, or RDoC, which was a multi-level, dimensional framework for thinking about mental health disorders. So it's quite different in its concept and its scope from DSM. DSM is a categorical system which doesn't include any biological data. RDoC and other frameworks are different both because they're dimensional, not categorical, and they include a hierarchy of different kinds of data, clinical, psychological, biological. I think that has to be the future, but it hasn't actually happened yet. And RDoC must be about 10 years old now. So there are very entrenched, vested interests, you might say, or reactionary forces, you might say. There is a lot of inertia around this. And it's interesting to wonder, well, what will actually catalyze change? I think the genetics is going to be very powerful when that finally kind of breaks through, because we know already that the genetics don't map on to DSM categories very neatly. Some of the risk genes for bipolar disorder are the same as some of the risk genes for schizophrenia, for example. They are not categorically distinct when you look at the genetics. The other thing that could break through is treatment. I think if we had a successful trial of an immunotherapeutic for a subgroup of patients with psychosis or depression, whatever it might be, but some kind of subgroup of patients defined ideally by a biomarker that predicted response to a new drug and delivered treatment benefits, that would be quite powerful, because that would demonstrate that actually thinking about how you diagnose an individual with a psychiatric disorder, whether you do that using DSM or you do a deeper dive and subgroup based on a biomarker, has implications for treatment. I think that could catalyze change.
Thank you.
Okay, one last short question and short answer, because we have to close.
Okay. Very quick question, although I'm not sure it's a short answer to these questions. But you moved very much between peripheral inflammation, central inflammation. The lady you showed at the beginning probably didn't have depression before she had rheumatoid arthritis, I'm guessing, and then you moved within the sex differences and epigenetics. So is this all coming to a head in terms of microglia and astrocyte activation pathways? Does it? Is there a common feature that's going to land? I sort of looked at the diagram you had at the end there that tried to give a commonality, or do you think it's going to be much more? I guess you're going to say it's much more complex than that. But how do all these things relate to each other, and how are we going to dissect all of that?
Yeah, well, that is a big question, and you're right. I don't have a quick answer to it. I just think so what I would say more seriously is the thing about the balance between peripheral and central. That's difficult from a clinical point of view, obviously, because we just don't have very good read-offs of central immune status. I think in the animal literature it's a bit clearer how the central immune cells are involved. But what I find exciting about it is there are still a lot of unanswered questions, but I get the feeling that this kind of approach ties up or begins to tie up a lot of loose ends and begins to offer us some more integrative model that is tractable and that we can pursue and experimentally refine over the years to come. And I think that's exciting.
Right. So it's my privilege to thank you again